All right, greetings everyone. This topic here for our Unit 5 heredity unit is all about Mendelian genetics. So hopefully this is a good review / refresher of Gregor Mendel and his famous or infamous, however you want to think about it, pea plants, and how he took his pea plants and crossed them with each other. He crossed them with themselves, with self-pollination, and had all of these different traits that he recorded for 10 years and 10,000 hours spending counting pea plants. And so we will not take as long as Mendel did collecting his data, but we will talk about some of his findings and what his conclusions can tell us about how certain traits or genes can be passed from parents to offspring. And so this first page is just kind of like a quick review or some background knowledge that we've talked about before. Information gets passed on from parents to offspring that is carried in our nucleic acids like DNA and RNA. That gets passed down from meiosis in our chromosomes during cell division, and that provides just a continuity of life. Over time, we'll start talking about like evolution and natural selection, and how over time maybe we'll see genes that have advantageous benefits to them, where they can maybe give them some ability to survive longer, will be found more prevalent in a population over time, versus traits that give individuals a disadvantage in an environment. And so we'll talk about that. So we have DNA and RNA, our two classic nucleic acids. We see double-stranded, single-stranded. We're not really going to be spending a whole lot of time with that on this unit. And so shared processes support concept of common ancestry. We'll see this pretty often. The idea that organisms across that don't seem like they're very closely related share the same processes. And so at some point, maybe that's evidence that they shared a common ancestor that used that same process, right? We'll call it like process A. And we see that like, oh, animals also use process A, and then bacteria also use process A. So maybe that points to some common ancestry that we would see way back when. And so some of that idea is that all organisms use nucleic acids to store and transmit genetic information. The fact that all of life shares DNA with each other is probably our biggest piece of evidence that all life shares a common ancestor at some point. We also have that all organisms share ribosomes, and they use ribosomes to make proteins. Also, all organisms have a cell membrane that's made out of lipids, right? So it's one thing for life to have the same basic functions, protection, genetic information, and proteins. The fact that everything is made out of the same components, the same macromolecules, is evidence that they have the common ancestry. And so where does this have to do with genetics? Well, the fact that life started at one point and has been passing on these qualities and traits from generation to generation over 3.8 billion years is pretty convincing evidence. And so we have these things that we can also look at. We have these like what we would call core metabolic pathways. We talked about these in Unit 3, cellular respiration and photosynthesis. Within those two things, mainly glycolysis is the one that is found in all life. All right, if we think about glycolysis, the most base version of it is you take glucose and you get ATP, right? So you take a food and you take maybe you don't necessarily need oxygen, but you just break that down into ATP or energy. And so all of life does that and can do that. All right, so let's get back to our main point of the notes for today, which is Gregor Mendel, and his laws that he talked about to describe the inheritance of genes and traits on different chromosomes. All right, and so the first thing we can talk about is an allele. So we have genes which are found on our DNA that code for specific proteins or traits. Alleles are variations of a gene. And so we get our alleles inherited from our parents. Organisms can inherit different alleles for the same gene. Right? This is where we get our big A, little a that we probably know and did Punnett squares with way back when. Right? And so we have a dominant allele. Always shows in the phenotype if inherited. Phenotype are these physical traits. These are what get expressed from the gene. Okay, think hair color, eye color. You could think organs, cells, whatever protein get produced. That's the phenotype. Versus the recessive allele, which when paired with the dominant alleles, does not get expressed at all. It is recessive. And so that is usually denoted by a lowercase letter. Dominant is uppercase. And so every organism, or organisms, we can determine or look at their alleles by looking at their genotype. So it's a combination of inherited alleles. So one from mom, one from dad. And so we have homozygous genotypes, right? Our prefix homo means the same. And so this is when you inherit the same alleles from both parents. So that can either be homozygous dominant or homozygous recessive. You can't just say homozygous. That is not enough information. You have to clarify. This is compared to our other type, which is genotype, which is heterozygous. Hetero means different. And so in this case, we're talking about two different alleles. One dominant, one recessive. Big A, little a. And so heterozygous genotypes always express the dominant allele. And then something we just talked about a second ago, our phenotypes are the expression of the genotypes, right? Genotypes are found on our genes or DNA. Phenotype is expressing that. All right, so Mendel had some conclusions that he came up with, right? He recorded and counted all of these pea plants, and he came up with these conclusions that we want to be able to apply to different situations. Now, these can be kind of easy to mix up, so I'm going to do my best to try to clarify when you can distinguish one from the other, and we'll look at some examples in class or through different assignments. And so the first one is called the law of segregation. All right, so this idea starts with the fact that, chromosomes, some chromosomes here, carry alleles. And so homologous chromosomes, this is in the meiosis topic, carry alleles for the same trait. They don't have to carry the same allele, right? We look at a homologous pair here. Maybe this is a dominant one, and maybe this is recessive. Versus this cell, maybe this is from mom and dad, and maybe this one is little a, little a. And so what we'll see is when chromosomes are separated, let's start this one, into daughter cells during meiosis, so law of segregation is all about meiosis, the alleles for each trait are also separated. And this is random, but this idea will allow for genetic variation to increase within the gametes, promoting genetic diversity. Okay, so Mendel's experimental design involved specific crosses with the P. We'll see some symbols here. P means parent. F1 is going to be the first generation from the parents versus F2 generations, which are like the grandchildren of the parents. And so we can use P, F1, and F2 to kind of talk about these traits that we see over time across generations. If we go back to our Punnett square that we'll see, we have these alleles that get expressed, little a, and then what we'll see is if we did like a cross, we can kind of see where our recessive alleles will go, where our dominant alleles will go, and this is basically where we come up with probabilities, right? So we have two offspring, each Punnett square are potential outcomes for offspring genotype, right? So of our four potential outcomes, two of those potential outcomes are offspring that would be heterozygous, right? So 50% heterozygous, 50% would be homozygous recessive. So 50% would express the dominant trait, 50% would express the recessive trait. But I think sometimes that gets lost in the in the terminologies and the Punnett squares is that these alleles are found on our chromosomes, our homologous chromosomes. Right, so we always want to think about, like, where is this coming from? What are we talking about? What is the context? So homologous chromosomes. All right, and so we can, if we track these P's, F1s, and F2s, we might be able to see some traits disappear, like a recessive trait disappear, and then come back in the resulting offspring or resulting generation. Okay, and then we can also take a quick look. Here we have a purple trait in flowers, which looks like the dominant trait or phenotype. Here the white phenotype is recessive, little r. We do our Punnett square cross, and then we can see we have all offspring inherit the purple allele or purple trait, right? And these are, again, that's too big. Right, this R allele is found on a chromosome here. Here's the other one. And then this one, maybe it should be the same type of chromosome, right? Like chromosome 1, like humans have 23 pairs, so maybe for flowers, this is a specific chromosome number. And then you take those offspring, these are all heterozygous, big R, little r, and you cross them. So we go from parent generation to F1 generation to F2 generation. And we can see, even though they both express the dominant trait, we can see that this recessive trait can pop back up, even though it appeared to disappear. Right, so you can still carry recessive traits without expressing them. And so law of segregation, you want to associate directly with meiosis and the idea that we are separating daughter cells during meiosis. All right, our next law is the law of independent assortment. And so this idea is that two or more genes assort independently of each other. Okay, so genes, the basic idea of this is that genes are not linked. We will talk about linked genes later, but this is going to be a key component that we'll see whether or not we can do a Punnett square and get expected results for them, whether or not they're linked or not. So one trait is not automatically inherited with another trait. They assort independently of each other. That's like saying, if they're not linked, the independent assortment would apply. That's like saying, if I have blue eyes, that means I'm also going to have red hair, right? Or if I have red hair, that also means I'm going to have my index finger longer than my ring finger, right? We know those traits are not linked, and so we are wanting to see the predicted outcomes of, oh, what's the probability of inheriting red hair and long index fingers versus blonde hair and short index fingers, and things like that. And then, so these alleles, again, on the chromosomes, for separate traits can be packaged in every possible combination into gametes. Right, so here's our, we have two homologous chromosomes. If we wanted to just do a quick review of like meiosis or ploidy number, this would be like 2n equals 4, right? It's 2n because we have homologous chromosomes. It's 4 because we have 4 chromosomes in here. And so from meiosis, right, this is meiosis. At the end of this, each one should be n equals 2. Right, we no longer have homologous chromosomes in each one, and there are 2 chromosomes. And so this is where we want to, again, keep thinking about these chromosomes and these genes that are found on them. But we can see that A and B have no relations to each other. These are the possible outcomes. These are also gametes. And actually, this is, when we do Punnett squares, this is basically what we're doing. We're just doing crosses, like a Punnett square cross to determine the probability of inheriting these traits. All right, so the next law is called the law of probability. So we can apply this to analyze single gene traits from parents to offspring and determine the possibility of inheriting certain types of traits. And so there's these laws that we can talk about that's found on the equation sheet for biology. And so let's try to figure out, to determine the probability of A or B happening, we have to add the probability of A to the probability of B. So let's say I'm flipping a coin, and let's say, what's the probability that I land a heads or tails? Well, we know landing a heads is 50%, or 1 out of 2. Heads or tails, so we have to take our B, tails, which is also 1 out of 2, 50%. We have to add them together. What do we get? We get 2 out of 2, or 100%, right? Because we just added our 2 probabilities or possibilities, right? There's only 2 sides on the coin, so getting a heads or a tails, well, we're going to get one of them, so the outcome of that is going to be 100%. Another idea is, okay, what's the idea if I roll a 2 on a 6-sided dice? Rolling a 2 is 1 out of 6. What's the probability that I roll a 2 or a 6? Then we just take our fractions, that's also 6. We add them up, that would be 2 out of 6, or 1/3, right? Because a 2 or a 6 is 1/3 of the dice. So we would expect 33% there. So if it's asking or, you want to add them together. We'll see this in genetics, like what's the probability of getting red hair or a long index finger? And then so you look at your Punnett square, you see the probabilities for both, and then determine that. All right, next, to determine the probability of A and B happening, you want to multiply the probability of A and the probability of B. So this is going to get like a lower, smaller number. So what's the probability that I roll a 5 and then a 6? So that would be 1/6 that I roll a 5 and then a 6. So you have to take times probability of rolling a 6, 1 out of 6 also. So the probability of rolling a 5 and then a 6 should be 1 out of 36, right? It's pretty unlikely that you're going to do 2 specific numbers back to back, right? And so what's the probability that I roll, or I flip a coin twice and I get heads twice? So a heads and another heads. That would just be 1/2 times 1/2. And so it's 1 out of 4 that we would expect to see that. And so for this to happen, we have to assume that genes are not linked. We'll talk more about that in a future topic, but that's basically how close are they on the same chromosomes, or are they on the same chromosomes? And so if the probability of allele A in a gamete is 1/2, like from a Punnett square, and the probability of allele B in a gamete is 1/2, what is the probability that both A and B are in the same gamete? So if it's and statement, right, that's where we multiply. So it would be 1/2 times 1/2. And so it would be 1/4 probability that the gamete is going to have both of those traits, or both of those alleles in a gamete or sex cell. And so we do Punnett squares to determine the probabilities, both or statements or and statements. And so a monohybrid cross is an examination of how one trait is inherited. A dihybrid cross is an examination of how 2 traits are inherited. A lot of the ones we'll see in monohybrid have 4 squares, right? You might remember that, where we do a cross. Punnett square, we're going to have 16 squares because we have a lot of stuff going on. We'll talk more about that later. And these Punnett squares that everybody loves really are typically used to illustrate the probable outcomes of a cross. So here we have a monohybrid cross. We have 1 trait. Here we're looking at flower color. 2 outcomes for flower color. We have purple, which is the dominant trait, and then we have white, which is recessive. So 2 possible phenotypes, but 3 possible genotypes. Homozygous dominant, big R, heterozygous, big R, little r, or homozygous recessive, little r, little r. And so we can calculate this as we need to. And so we can take this information and we can do Punnett squares, but we can also do this for like family history. And this is how we can group family history, and we can predict or determine possible inheritance of traits and genes. And this can become important when we look at like diseases or recessive disorders. And we can determine what's the likelihood that an offspring inherits this disorder. And so a pedigree, the visual representation tracing the history of a trait through familial generations. Like I said, this is going to help identify modes of inheritance. There are more than one way, really more than two ways if you were just thinking dominant and recessive. There's a few other options out there. In any pedigree, we have circles represent females, squares represent males. I always think about SpongeBob. SpongeBob is a square. And then I think about Sandy Cheeks from SpongeBob. She has a helmet on, and so she's a circle. SpongeBob's are squares, females, circles, males, squares. And so shaded shapes are affected individuals. So that whatever trait we're interested in could be dominant, could be recessive, could be sex-linked dominant, could be sex-linked recessive, could be linked genes. But whenever we do that, we shade those in. Perpendicular lines represent offspring. That generation can be indicated with Roman numerals. So maybe this is Roman numeral 1, this is Roman numeral 3 or 2, and this is 3. And sometimes we can say, you'll see it referenced as example 3-2. That means the second individual in the third generation that would be talking about this male here that's affected with some type of trait. Okay, so a few ideas. We have basically we have these things called autosomes, and so autosomes are chromosomes 1 to 22 in our nucleus, in our cells, and so these are also called like body chromosomes. Okay. This is different from sex-linked chromosomes or sex-linked traits. We have sex chromosomes, which is the final pair, 23. If you remember, males have the sex chromosomes XY, females have XX. Okay, so you can be an autosomal dominant, which is found on the body chromosome or chromosomes 1 through 22, or you could have a sex-linked trait found on chromosome 23. So you have autosomal dominant, you have autosomal recessive. We'll take a look at some examples here in class, but we can see maybe autosomal dominant. We don't really know what these genotypes are, right? We know if they're affected, they're going to have a big A here. And then we have to do some reasoning out, right? If they're not shaded, then they have to be recessive, right? And so if they're recessive, they have to have the same genotype of little a, little a. And so this one, we always want to like determine if we can figure out genotypes. And so the fact that this is not shaded is telling us that it has to inherit a little a from both parents, which means both of the parents have to be heterozygous. Does that mean we know what this not allele? No, we don't know if it's big A or little a, because we would need them to start having kids to maybe start making some predictions. But with this information, we don't know that that son's the genotype. So parents have to be heterozygous in this case. If we look at another example, autosomal recessive. Now let's say for the fact that we don't actually know, and we have to guess, looking at this pedigree, is it recessive or dominant? This is where we kind of play the hypothetical game. So if we thought it was dominant, maybe or excuse me, if we thought the shaded one was dominant, we can see pretty quickly where would that come from, right? Because if any of the parents had a dominant allele, which they need to because it has to come from mom or dad, they would be shaded in, right? We would need one of these to be shaded in to pass on that dominant phenotype. But the fact that neither of them are should be telling to us, right? We want to kind of play it out. Right? This has to be recessive because what's going on here is the parents have to be heterozygous because they have to express the dominant trait, but they still have to carry that recessive allele. Same thing with the male son again. We don't necessarily know their genotype. We know it has a dominant allele to start. Okay, and then we'll have sex-linked traits. So there's a lot of traits that can be found on sex chromosomes, specifically the X chromosome. So the X chromosome kind of looks like a normal chromosome. The Y chromosome, and there's all these different alleles, all these different genes on here for different alleles. Right, A, B, C, D, E. Like all of these are different genes. Versus the Y chromosome, it's not a lot going on there. There are traits that can be found on the Y chromosome. It's not a ton, but they would be separate from the X chromosome. And so sometimes we'll talk about sex-linked traits in the next topic. It's more complex patterns of inheritance, but we can use this to also determine how offspring are inherited. So we'll take a look at this maybe more in class. We'll take a look at some check-in questions down below, and I hope that was helpful. Thank you.